Hello, everyone. I'm Edwin. Today, I just want to briefly talk about what my lab is doing. We are doing the neuroimmunology research. What that means is we study the crosstalk between neuroscience and immunology in the context of several neurological diseases. Here is my email. If you have any questions, feel free to email me. And so students in my lab can either enter the immunology or and microbial pathogenesis program or the neuroscience program. And currently we are mainly doing studying two diseases. One is post-stroke brain inflammations and recovery, and another one is multiple sclerosis. Um, so obviously we cannot do research in human brain, so we use animal model to do it. For stroke, we do medial cerebral arterial occlusions, and for multiple sclerosis, we use experimental autoimmune encephalomyelitis as a model. So both diseases involved um, the interactions between the immune cells in the periphery and the glial cells in the central nervous system. And we are interested to look at first how brain recovery following ischemic stroke um, is regulated by the interaction between glial cells and immune cells. And second, in terms of multiple sclerosis, we want to look at how the immune cells and glial cell interactions promote the progressions of multiple sclerosis, which is an autoimmune disease. Um, so technique we use in the lab, we have multiple transgenic and knockout mouse strain, um, and we do functional analysis, um, functional study and cell analysis ex vivo. And we also do a lot of histopathological studies, immunofluorescent imagings. We use a lot of flow cytometry to look at immune cells profiles, next generation sequencing. And what we are trying to develop now is behavioral studies of the animals. And the pictures here is just one of the example. What we are doing is a project of one of my PhD students where we study the role of interleukin 34, IL-34 in brain recovery following stroke. The IL-34 is on white here, which is produced by neurons in purple. So here we are looking at a brain slice where the mouse was ischemic stroke is induced in the mouse, and we look at the brain, and this is the scar area of the brain, and we see green microglia. They are resident glial cells in the brain, and they start to take up dead neurons. As you can see here, you have purple neurons taken up by the microglia, and here we see a microglial cells taking up a dying neurons. And as we want to study how the peripheral immune cells involved in this brain recovery, we also look at red cells. They are monocytes from the blood going to the brain, and you see here they're interacting with the microglia. So this is one of the studies that we are having now. Um, the lab atmosphere. So we kind of a small group, and currently I have two PhD students, one research assistant, and a couple of undergraduate students working in the lab. And I would say that we are pretty close to it, working close together. Everyone have their own projects, and also they help each other. So if you want to learn more about the lab atmospheres, feel free to come to the lab and talk to the students. And in terms of the mentoring style of me, my office is within the lab, so I see my students every day, and we talk about our projects every day. And my experience in mentoring students is that the first or second years, uh, students usually work closely with me and on experimental design and learn techniques from me and other students directly. But the students are expected to develop independency during the progressions of their PhD studies. And so far, my two PhD students somehow has managed to develop new techniques for the lab that um, uh, I don't know how to do it. So they develop it. And they teach everybody in the lab. So it's kind of uh, this is kind of my mentoring style. So if you are interested to learn more about the lab and potential rotation opportunities, feel free to contact me.